This is one of the toughest problems you've had so far in the course. And let's break it down one step at a time. You need to figure out the double-stranded DNA sequence, the mRNA sequence, and fill in the blanks for the amino acids in the resulting protein based on the limited amount of information that you're given here. One of the first big questions, conundrums that you have to figure out is which of the DNA strands of the double-stranded DNA is being used as a template to make the RNA from, to then go on and make the protein. And there's only one clue in this entire diagram that gives it away. Here, the T, thymine, and the U for uracil. Remember that both thymine and uracil base pair with A. They are interchangeable with one another, T and U. So if the mRNA sequence has a U here and this strand of the DNA has a T, we know that the other side of this double-stranded helix must have an A based on our DNA base pairing rules. And A pairs with U, which means that the mRNA sequence was transcribed by pairing with the top strand here of the DNA double helix. So let me just fill in a couple of blanks here. Guanine pairs with cytosine and adenine pairs with thymine. And now we know that the uracil paired with the adenine. So to fill in the mRNA sequence, you need to know what's complementary with cytosine and thymine. Well, here we know it's G, and here we know it's A, guanine and adenine. And notice that the mRNA sequence looks identical in this codon to the DNA sequence from which it was not transcribed, right? The non-template, right? TGA, UGA, they're the same, it's just that the thymines have been replaced with U's. So let's see if we can fill in the rest of the pieces here. We know that guanine pairs with cytosine, thymine pairs with adenine, cytosine pairs with guanine. But how to get these other pieces? Well, we know that cytosine pairs with guanine, and we know that the guanine, what it pairs with goes here. This sequence of DNA should match the mRNA sequence exactly, except for the thymines and the uracils. So I can put those pieces in there, and I can fill in this piece here. So now we've got the DNA double helix down, but we're missing this one codon here. There's only one codon for tryptophan, and it's UGG. So this means that the sequence here in the DNA would be TGG, right? Because it matches the mRNA except for the T's and the U's. And here we'd have the complementary sequence ACC. Now we have the complete DNA double-stranded sequence. Now let's fill in the mRNA. We know that's going to be a guanine. It's going to be a cytosine and a thymine, right? It should match here, except, I made a mistake. My mRNA has a T. It should only have a uracil, right? All thymines are replaced by uracils. So now let's fill in the protein sequence. GCA codes for alanine, or ALA. We already have tryptophan, and look, UGA is what? It's a stop codon. So nothing happens here. This is a stop. There's no amino acid. And then this codon, CGU, it codes for arginine, but do I write arginine here? No, because it's after the stop codon. Nothing's going to be translated here because the ribosome fell off at the stop codon and translation stopped. So hopefully you were able to arrive at this answer by synthesizing all of the rules and the information we've learned about in lesson 2 and 3 up to this point.